So I'd like to welcome everybody to our student award talks. And first of all, I would like to read our land acknowledgement and just introduce a little bit about the CRSC. The CRSC, the Color Research Society of Canada, activities related to sharing color knowledge take place across Canada, understood as part of Turtle Island, the ancestral homelands of over 630 First Nation communities, representing more than 50 Indigenous nations and languages. We work to respect, affirm, and support, where necessary, the inherent treaty rights of all Indigenous people across these lands and water. Our commitment to honoring the knowledge, ways of knowing, and wisdom of Turtle Island's Indigenous peoples aims to contribute to the decolonizing work required today for moving towards a just society. So I'd like to welcome you to tonight, if you're in Toronto or maybe you're somewhere else and it's morning, talks, our student award talks. We are, as I mentioned, the Color Research Society of Canada, the Canadian member of the AIC, the International Color Association. And the CRSC is a nonprofit organization for color research focused on a cross-disciplinary sharing of color knowledge. We seek to develop and support a national cross-disciplinary network of artists and designers, scholars, and practitioners with an interest in engagements with color, and to encourage discourse between art, sciences, and industry related to color research and knowledge. And we started our student award program this is the third year that we've had awards, and we're very happy to, I'm going to tell you a little bit about the student awards in case you want to send it on to others that you think may be interested, other students across Canada. And our student award is open to all post-secondary students, so graduate, undergraduate, cégep, studying in Canada or recently graduated. And the focus of their study is color in any field within 18 months of the submission date. And our submission date is coming up March 1st, 2023. So color must be the main topic of the study, but all disciplines or a combination of disciplines are welcome. Research-based or practice-based studies are welcome. For example, science, social science, history, literature, art, design, architecture, film, and individual or group submissions are also acceptable. So I'm very happy to present diverse awards from this past year, because we have science and we have art represented. And we are starting our talks with our graduate award recipient, Paria Mehrani. So she completed her thesis at York University, and she will be talking about a biologically inspired neural network for color representation. She is currently a postdoctoral fellow at York University, where she focuses on active model learning in reinforcement learning. Prior to her postdoc, Paria was a PhD candidate under the supervision of Dr. Tsotsos, where her work included developing biologically inspired artificial systems with a focus on computer vision. During her PhD, she introduced analytically defined neural networks designed according to known neural mechanisms in the brain. Her interdisciplinary work at the intersection of computer vision, machine learning, and computational neuroscience serves the dual purpose of introducing automatic vision systems and suggesting models that promote our understanding of the brain. She has published widely, and she was awarded her PhD in electrical engineering and computer science from York University in 2021. I'm very happy to welcome Paria, and I will just set you up to share the screen.
Thank you.
Okay, we should be good to go.
Great. Now you see my screen, my full screen?
Yeah, all good.
Okay, great. Thank you, Robin. And good evening, everyone. I'm so excited that I'm going to talk about my work that revolved around the question of how our brain sees color. This has been a puzzle for scientists because color does not exist. There is light of different wavelengths that our visual system transforms into various perceptual hues. These perceived perceptual hues are contributing features in many vision-related tasks. For example, if you are looking for a yellow rose in a colorful rose bouquet, color helps us decide which stem to pick. Therefore, understanding how the brain transforms light to color will promote our understanding of its visual processing. Additionally, revealing color processing mechanisms in the brain can help in designing artificial vision systems for tasks which color is a prominent visual feature. For example, when our goal is to segment an image such as this into two regions, flower and the ground. So to address this problem of hue representation in the brain, we introduced a biologically plausible computational model that we called HueNet. And computations in the various components of this model are inspired by our current understanding of visual processing in the brain. HueNet then suggests mechanisms that help in explaining the observed color representations in the brain that I will tell you more about later. And I must clarify here before I get into details that our focus was on hue representation, leaving modeling other color components for future work. But in this presentation, I will use hue and color interchangeably. But the question is, what do we know about hue processing in the brain? Well, we know that color vision in humans start with 3 cone types in the retina, sensitive to long, medium, and short wavelengths. Then visual information in the form of activation signals from these cones go through the optic nerve and then go through a hierarchy of processing stages. Neurons in the first processing stage, the LGN, have single opponent receptive fields. I have an example of a single opponent cell here where the cones sensitive to long wavelengths excite the neuron and they send their signal to the center of the receptive field of the neuron. And its responses of this cell is inhibited by M cones, shown as green triangles here, on the surround of its receptive field. Therefore, the best stimulus for this neuron will be a red disc that will activate this part, but does not activate the M cones on the surround. Neurons in the LGN are strongly activated when hues along these 2 axes that are marked with black X's fall on their receptive fields. This is the McLeod-Boynton color space with red versus green and yellow versus blue axes. Then LGN cells send their signal to single opponent neurons in V1, where a rectified sum of the 3 cone types can model single opponent V1 responses. Moreover, V1 neurons are selective to hues along these 2 cone opponent axes, as well as to intermediate directions with some examples that are marked with red X's here on the right. The next processing stage after V1 is V2, where a higher percentage of nonlinear neurons were reported in this visual area with narrower tuning bandwidths. This figure that I have on the right depicts what I mean by narrower tuning bandwidth. Suppose that we are recording a neuron in V2 and we plot its responses as a function of the different hue stimuli that we present to this neuron, then this function will usually look like a bell curve. And then the width of this bell curve represents the tuning width. For example, here the orange curve has a wider tuning width and the blue one has a narrower one. This tuning bandwidth will tell us the range of hues that can strongly activate the neuron. With a narrower tuning bandwidth similar to the blue curve here, we will have neurons that are more selective, which will eventually result in better discrimination of slightly different hues. For example, this blue neuron with the blue tuning function can better discriminate between green hue and cyan-like green hue, as the difference of responses are larger here on the blue curve. Alongside the high percentage of nonlinear neurons in V2 and also narrower tuning bandwidths for V2 cells, these neurons were also reported to have a diverse set of selectivity, specifically to intermediate directions, intermediate hue representations that are shown by these red X's here in the MB space. Then V2 neurons send their signal to cells in V4, where clusters of hue selective neurons were with similar hue selectivities were found. What we have here from a study from 2014, they recorded V4 area in macaque monkeys, and we see here patches of clusters of neurons. Each patch is represented by a dashed line with a form of a square, and within each patch we see some colored curves. Each colored curves is a cluster of neurons in V4 with same selectivity, and within each patch we see a variety of hues going from green, yellow, and so on. So a variety of selectivities were identified in this visual area V4. So color processing in the visual cortex goes beyond V4. However, in this study, we limited ourselves to modeling up to V4. Therefore, I will stop here on the biology side. But to summarize what we know about hue representations in the brain, we know that with 3 cone activations, color vision begins, and the signal goes through a hierarchy of processing stages. Neurons become more selective with narrower tuning bandwidths, and intermediate hue representations start to develop throughout the hierarchy as we go higher up in the hierarchy. Given these observations about the brain and what we know about processing of visual signals in the brain, our computational models have not moved past single opponency in LGN and VIVAN, which leaves us with the challenge of explaining the observed representations in V2 and V4, specifically narrower tuning bandwidths of these neurons, and also the diverse hue selectivities in these visual areas. And to address this problem, we introduced HueNet, whose architecture is shown here. And what I mean by a network or net is that similar to the hierarchy that we observed in the brain, our model has different stages or layers of processing that explicitly model neurons in each of those visual areas, LGN, VIVAN, V2, and V4. Neurons in this network in HueNet are implemented according to experimental findings in each of those visual areas. For example, LGN and VIVAN neurons have single opponent receptive fields, and we employed a half-wave rectification function to model responses of single opponent VIVAN cells, as was reported in biological VIVAN neurons. But the important contribution of this network was the introduction of neurons that we called multiplicative cells that help in explaining the observed representations in V2 and V4. Specifically, we proposed that narrow tuning bandwidths and also selectivity toward intermediate hue directions that were reported in V2 and V4 cells can be achieved if we use multiplicative modulations. That is, we multiply activations of V1 neurons that feed to these cells in V2. Then finally, in our last layer of the network, in our V4, responses of single opponent V2 cells and multiplicative V2 neurons are combined to model neurons selective to 6 different hues, ranging from magenta all the way to blue. And the angular position of these 6 hues are shown with the letters on the unit circle of the MB space here. Now, to wrap up HueNet, here's an example of model neuron activations to the hue wheel that is shown here on the left. This is the input to our network. The arrays that you see within each layer represent maps of neurons with the same hue selectivity. So if we go to the very last layer here, V4, this is the map of neurons that are selective to blue, for example. Each map, the responses of the neurons in each map are shown in grayscale with lighter, stronger responses as lighter values. Now, if we look at this model layer 2, we have the multiplicative modulations represented as X's between the 2 types of single opponent cells whose activations were multiplied to achieve selectivity in those multiplicative cells. In our model layer V4, the 6 types of hue selectivities are shown. And we see here that the light area in these maps sort of rotates around the hue circle from magenta to red and then yellow and so on, meaning that neurons whose receptive field overlap with those particular hues in the hue wheel are strongly activated in those maps. Now, in order to test the performance of this model, we recorded neuron activations to 60 sample hues from the HSL space. The dots that you see here on the unit circle of the MB space represent those sample hues, each dot for 1 sample. The gray curves in these polar plots represent the tuning functions. Those bowl curves that I talked about earlier are now shown in this polar plot, in this polar color space. What we see here on the left is the tuning function of a single opponent neuron in our model layer V2. And on the right, we have the tuning function of a multiplicative cell from our model layer V2. Comparing these 2 tuning functions show that the tuning bandwidth of our single opponent cell is wider compared to our multiplicative neuron, and also that this neuron peaks close to a cone opponent axis, whereas the peak for the tuning function of the multiplicative cell is shifted toward an intermediate hue direction. Now, in order to quantify those observations about the tuning functions, I computed the tuning bandwidths of single opponent and multiplicative model V2 neurons and plotted the tuning bandwidth distributions. Here, the light blue and light orange curves show the distribution of tuning bandwidths for multiplicative and single opponent model V2 neurons, respectively. And we observe the shift in the tuning bandwidth distributions from single opponent cells to multiplicative neurons, suggesting that the overall tuning bandwidth in multiplicative cells were narrower compared to single opponent neurons in our model. This shift in tuning bandwidth was also reported in biological cells that were classified as nonlinear and linear in visual area V2. Then I evaluated the distribution of selectivities in the various layers of hues, specifically the hues that strongly activate neurons in each of those layers of the model. On the right, you see a polar histogram of peak selectivities, the peak of the tuning function for all neurons in all layers of the model. Whereas the single opponent cells peak at hues that are sort of close to those cone opponent axes, multiplicative modulations further shift those peak selectivities to intermediate directions. In other words, we observe that multiplicative modulations, with those modulations, the representation of hues along intermediate axes start to develop in our visual hierarchy. And as a final note on HueNet, at the beginning, I mentioned that having such models that are biologically inspired, we can also use them as computational vision system that can be employed in performing visual tasks. Here, I have shown some examples of how we can use the model V4 representations in a flower segmentation task, demonstrating how those representations can be employed in a vision system that is to perform flower segmentation. These on the left side, you see the input image, and on the right side of each image, you see the segmentation. For example, here the blue one shows the how the model segmented the flowers versus the background. This image, however, is very interesting. That the model segments the whole flower in one piece, even though we have shadows on the flower here in the input image. Here the hue is constant, but the overall appearance of the flower color with the shadow is not. However, our model could segment the flower as a whole. Now, to summarize HueNet, our goal was to suggest hue processing mechanisms that can explain the transformation from cone opponent representations to a diverse set of hues in higher visual areas, specifically V2 and V4. For this purpose, we introduced a hierarchical model that explicitly models neurons in each of LGN, V1, V2, and V4, with neurons implemented according to experimental findings. This model shows how we can move beyond single opponency to model neurons with narrower tuning bandwidths and representations along intermediate hues with a diverse set of selectivities, similar to what was observed in biological hue selective neurons in V2 and V4. Multiplicative modulations that were introduced in our model were key to narrower tuning bandwidths and the shift in tuning peaks toward those intermediate directions. Our work also suggests that the brain's neurons also employ such multiplicative modulations, which result in the reported nonlinearity in their tunings and shifting of tuning peaks to intermediate directions that together would result in better discrimination of slightly different hues, such as red versus orange, and representations of span color space. And finally, we demonstrated that HueNet as a computational vision model could be employed in a vision task with color as a prominent feature. Here, we had the example of flower segmentation. And on that note, thank you for your attention.
Thank you so much, Paria, and congratulations again for your award. Please join me to clap virtually, or you can put your camera on. Yes, thank you. Brian, could you stop share and I'll take any, does anyone have any questions or comments? You can put them in the chat, or you can put your hand up and unmute.
I've got a couple of.
Okay. First, Montaha has a question from the chat. Is HueNet considered an AI?
This is a very interesting question. That's the ultimate goal. Overall, all the models that I worked on can be used as part of a bigger AI system. This is just one. So AI is so big. These models are only considering vision. And this model that I talked about, HueNet, is only considering color processing. But yes, this can be a component of a bigger AI system. Yes. V3 is interesting. Yes, there is a V3 area in the visual cortex. Overall, we say that the visual cortex has two main streams to that, or like two branches, where the visual signal travels through. There are many interactions between these streams, but what I presented in HueNet were the major areas in the ventral stream of visual processing, which is believed to be doing color processing in the visual cortex, although there are studies that say that the dorsal stream also do some sort of color processing, but the neurons in this stream are more sensitive to color. And V3 is like going back and forth between these streams. So yeah, the brain has a very, very complex architecture. V3 is somewhere in between, but this was the main flow of signal in the ventral stream that I presented in this work.
Okay, thanks.
No problem.
Brian, do you want to just ask your question?
Sure. Hi. Interesting talk. Thank you. I got a couple questions. First of all, how stable is it under variation and illumination? I mean, I'm not asking for strange illuminations, but just daylight versus different times of daylight. Do you get the colors, or what happens?
We didn't model for that. We didn't. It was just like pure hue. Even yeah. So the samples that we had for from the HSL space had fixed saturation and lumens. So it was just the hue that we varied in all those samples that we had from the HSL space. There are many different aspects of color processing in the brain, and we just focused on one aspect in this project.
So you just took in different kinds of hue, as opposed to you weren't taking like images and saying, what is this hue? Or I thought you were, in some sense. You showed like that color wheel, and then identified the hues in the different sections, but basically just 6 hues. But
yes.
At some point you talked about 60 hues. I mean, the finer separation. I mean, if there were, am I right? There were 60 hues, something like that?
There were 60 hues that we presented to the model, and we recorded responses of neurons in the model. In the very final layer, V4, we modeled only 6 neurons sensitive to 6 hues.
Okay, I understand. And there's no machine learning in this, right? You built the model. Is that correct?
That's correct. That's actually correct. Every aspect of this model was designed based off of experimental findings.
Sure.
No parameter was learned. I have another work that I did a little bit of learning in the very last layer of the model, but not in HueNet.
Okay, and then the crucial thing in HueNet is the multiplicative. That's the basic difference. Okay, as opposed to subtractive.
Yes.
Opponent here, multiplicative opponent or something. I don't know. Anyway, so that's interesting. I haven't. And is that your idea, or does that come from someplace else? Does it have a history?
No, that was my idea. That was my idea when I started working on this, and then later on, we experimented a little bit with pooling. So suppose that we have neurons that are selective to single hue, and they are in local neighborhoods. We start to, so each neuron looks at whatever neurons are in its neighborhood, and then it either increases or decreases its belief in seeing that particular hue based on what they are seeing. That could also result in narrower tuning bandwidth, but we concluded that that mechanism cannot shift selectivities to intermediate hues.
Right. Oh, interesting. I mean, I haven't heard anybody talk about multiplicative before, and it seems so, in hindsight, so obvious, right? If you can subtract, you can multiply. But okay, I lost my train of thought a bit. I did think it was a good idea. Oh yeah, do you have any physiological idea how the multiplication is implemented in the brain?
So in the paper that we published, it was published in Scientific Report. We cited some references that had found evidence of multiplicative modulations in the various visual areas. So we thought that if there is evidence of such modulations in the brain, why not with color sensitive neurons? That was where we did it.
Sure, no, I understand. I mean, it seems simple. It's got to be able to do multiplication somehow, but it okay. But it's not the sort of typical synapse thing, but okay. Am I going on too long? Somebody else?
There, one more. Yeah, we should move on to Annie, unless it's really quick. Montaha has a question in the chat, and Priya, maybe you can answer in the chat.
Move on.
Yeah,
we can come back to this. We can come back and have a discussion after. So I'd like to introduce our second award winner for 2022, our honorable mention, Annie Toole. She's at Memorial University of Newfoundland, and her work is entitled Mornings at the Press. Weaving and natural dye are at the core of Annie Toole's transdisciplinary studio practice that extends into printmaking, clay, and sound exploration. Weaving and its systems of notations, model translation, translanguaging, and writing. Through materials, she manipulates language and its forms. Failing to model the vibrations in space of simultaneous translations, her studio research speculates dual spaces that accommodate two languages at once. Currently based in Quebec City, she holds an MFA from Memorial University of Newfoundland, a fine craft diploma in constructed textiles from La Maison des Métiers d'Art du Québec, and a BSc honors in mathematics from the University of Ottawa. I would very much like to welcome and introduce Annie, and let me just let you share screen. Okay, it should work now.
Okay, you can see my full screen right now?
Yep, everything's good.
Yes. Okay, great. Well, first, thank you very much. And thank you, Paria. It was fascinating. And I'm very prepared. Yesterday, I was in a talk with the Catskill Weaving School, and it was Eric Hibbett presenting, he just wrote color theory for dummies. And so like, I feel like I was much more prepared and like better able to follow along. And it was really like good timing because that was last night. And I mean, he's a painter and doing it to artists. So I found it really interesting and was like just able to make parallels. So thank you. So I just want to thank the Color Research Society of Canada. It's been great to discover other color enthusiasts. And I'll also mention that I'm currently located in Quebec City, which is unceded territory of the Huron-Wendat Nation. And all right, so I'll begin by doing my round of thank yous. This project started with my MFA and has continued on since. So I'll be presenting research along the way. And so the Maison des Métiers d'Art, where I went to school and have continued weaving there since, the Aramont School of Art and Craft, the Conseil des Arts et Lettres Québec, and the Canada Council for the Arts. I received social science and SSHRC funding, the Memorial University, the City of Quebec, the Wingate Charitable Foundation in the US, and the Culture Capitale Nationale and Chaudière-Appalaches. So my talk will focus on the artwork that I presented called Mornings at the Press, but I'll begin by just talking about a bit about my regular artwork because it'll sort of just add context. This work that I did and presented was very different and a different approach to my normal working. And it's been very meaningful. And it also set me on a path post graduation. So this is called Connecting Band. It's a collaboration with Katarina Spikskam, who's a Sami artisan in Sweden. And it was presented in an exhibit called Shared Terrain at Harbourfront Center during Design TO last winter. And so these are all, it's a 30-meter woven band. Katarina weaves small traditional bands in heritage, and then we were looking at their specific looms. And so all of the dyes, I dyed the wool from New Brunswick and PEI, and they're all local colors from a grower of natural plants in Kamouraska. So all from the land where I am. And then there's a reindeer tassel from the herd of reindeers of Katarina. And so color is very central of making color and the materials in this piece. And then here I have sort of a trifold blanket, which is text, the translated text and the black translated that flips over. The wool is very bare and there are just accents of color. And I'm pointing this out because if color appears in my work, it has to be made. And for a long time, I mean, weaving and the structure is so central to everything. They're just details with different plants. And then also here, there's their trilingual dice. They're sort of like the keyboard, which is in French, English, and Spanish for me. Comes with sound. Again, they're bare. And I've been wanting to revisit these with color. It's been on my mind for quite a while. And sometimes there's just even absence of color in my work because I'm so concerned with other parts. This work is only pixels, so it represents the structure of weaving and creating associations with language, with naming, with recognizing where you are. So the notation is always very important. And so it's just using the basic pixels. And the same way sometimes is the reverse. I do ceramics and I became so fascinated when I had access to creating glazes that I could no longer think of form. And I'm still stuck with just color and not able to marry it with form because color is so pertinent or so like the inquiry is so big, I can't marry it. And so I thought I'd take you through the printmaking or the natural printmaking. So I'm a competent weaver and dyer. I've done a lot of that. And I came to printmaking because I wanted to print my weaves and I wanted to do it with natural dyes and pigments. And I think I didn't know where that would take me and how it might be complex. So these are my first tries. This is during my first residency. I did a low residency MFA in Newfoundland, and the print technician, Robin, I came with my little jar of madder pigments, and we tried a lot with a block, and it's a satin weave structure that I had carved into a block. And so we tried a bunch of different bases that we could use around the print shop. And then Robin told me, she's like, oh, you're a textile artist, you will love printmaking. And she was not wrong. And so this is sort of all the products we took out, and there was nothing, didn't find a product on that first try, and sort of knew that I wanted to look further with that. And so I went to Aramont School of Arts and Crafts in Tennessee. Before leaving, I wove a lot of fabric on the jacquard. I really came to printmaking, and I wanted to use my weaves to print with as a translation, as another way of seeing them. And that's also pushed my weaving in a whole other direction, because I became interested by what the fiber looks like, not when I weave it, but what it looks like in properties once I squish it, and once I squish it with ink. And also looking at weave structures that won't necessarily be interesting as cloth, but that will be interesting once printed. And so these are traditional inks, and they give me headaches. They're very, very strong, and a lot of people love the odor of the print shop, and it does not agree with me. And anyhow, if there's color in my work, I have to make it, and I need to have like a long-term built relationship to it. So for my MFA work, I'll just go back. For my MFA work, I wanted to do some prints and have them be, or take that time to develop new horizons and really look at making prints with natural dyes. Early in the pandemic, probably in the beginning, the first moment where once everything was closed, things felt normal, I assisted in an online Zoom class, maybe the first one, called Botanical Ink and Paint Making with Flora Arbuto in the UK. And it was very exciting. I learned to do lake dyes, and I was dyeing a lot of fabric, and I used to just pour out my dye baths when they were done in the drain. And there was all this color that I wasn't taking. So then once I had dyed my fiber, I could do a precipitation and then gather it, thicken it, it becomes a pudding, and then dry it, and then grind it and make pigments. That was very exciting, and I had a lot of dyeing to do. And so I quickly, this is like the following week, I have buckets everywhere. And those are my first ink samples. I started filtering them, and they got better as it went along. And this, I quickly made like a little kit for one of my children. And these are little pipettes of modifiers. So there's iron oxide and an acidic and an alkaline, and he can play with them. So everybody was also having fun with the natural color. And then I was working on an artwork called, it was for an exhibit called Will of Iron, and all on the theme of iron. So in craft and visual arts, but the material or taken in very diverse ways. My proposition was to look at natural dyes and how they're saddened and darkened with when using iron. And so it was to weave a large piece and have different areas that were all mapped out, and some were dipped for different amounts of time. And basically it was sort of looking at the social fabric on how at times there may be like harder moments, times that are darker. They may last a different amount of time, but taken as a whole in a textile, we can barely see it. I mean, the whole remains colorful and supportive and sort of blends it. And so I was making all of the natural dyes, and then I was also keeping this map of how long and which section had the intervention of darkened color. So I made like a register, and this is the artwork. And on the right, there's a large jar which has every paper for every intervention that occurred. So it's sort of taking the fiber back. For the weavers, it is a deflected double weave. So it's like two fabrics that are stuck together, and then there's two bands. And again, you see the papers and the percentage, like the concentration, the amount of time, and which it was. So it quickly became incorporated into my artwork. And then I tried my hand at making watercolor. I still have that. And I mean, there's a lot of trial and error. And this was the hardest watercolor, and I think it doesn't dilute. And I just keep the little container around as a reminder that sometimes you need to get some help. And there's not a lot. I found ZMA's printmaking, which is a wonderful resource in Massachusetts. For 20 years, they've been developing safer, ecological, people go from everywhere to train their ecological printmaking for traditional printmaking. And then they during the pandemic put on videos, and I took this concentrated dyes and monotypes, and my pigments aren't super well grinded, but it already got me further in. And this is taking the same lake pigments that I had, drying them on plates. I use a printing press, which isn't in my studio, so I would carry those and carry my wet paper in the street and go and use a printing press to transfer it. And so then I tried dyeing papers, and they're not very dark, and I wanted them really bright. And just looking through, because there's very little documentation on like hands-on printmaking and where you can really get good results. And it was hard to access the library too at this time. I was in a different city and it was the pandemic, so it kind of really shifted the path to that. But there's Radha Pandey, which does unbelievable research in Indo paper making techniques with natural dyes and paper. And so then I learned about brush dyeing paper, but she does book arts. And so when you're doing book arts, you're not wetting the paper, but already you can see that my papers are much, much darker. And the oranges matter. The right, I think it might be cochineal. And then this is sort of combining the two techniques, the monotype on top of the paper that's dyed. And these are just how I'm mixing my pigments. I was trying different bases. There's indigo and iron oxide, which are my favorite color choices, and they remain the ones that I grab. And as you can see, the texture is a bit ruddy. It's not, I mean, I'm trying to develop products that I enjoy working with, that I have a link with, but also that will give me good printmaking results. And the whole point of this artwork, Mornings at the Press, I gave myself a frame. I mean, I took my MFA to really look at my own relationship in the studio. I was reading on journaling, and at one point I fell on someone who's saying in his journal, he does not quote, the ideas have to be his own. And then I wanted to look at what type of writing would get me as close to the studio as possible, where things are not working, where you're trying to figure out things, and how that advances, and like what is the struggle, and how do you write that, and how do you make someone feel what's going on while you're working in the studio. So I really wanted writing to be close to the studio. And in weaving, it's very different. So with printmaking, I have a lot to learn, so it's perfect because I'm a lot more conscious of it. And so I gave myself a framework, and I was to go to the print shop. I was like, I'll do like a small edition. I will go to the print shop for like a month and a half, a couple mornings a week. I reserved a press, and I was like, I'm going to experiment. And from the experimentations, I'll decide what paper size I want, what technique I want, and then like I'll buy proper plates of the proper size, and I'll come up with a format. But as this went on, I didn't want to develop a perfect print with a perfect order to repeat it, to have it be perfectly made. The best parts, and what became the most interesting, was I would see like one paper would bleed, so I combine it with another paper and would layer them, and then would sort of like print off of another print. At the end of the session, there's leftover ink, so I just sort of like mix them together. And it's sort of those accidents that I want to provoke. And this really left me stumped for work because I was like, how do I present it? And do I want to present it? Is it ready? Do I present all of it? Do I present none of it? And it was really like provoking me. I don't necessarily get to having so much feeling about my work. And I was like, this is interesting. And so then I started asking people around me, and they were like, they had the same reply. They were like, maybe, maybe not. Like, I don't know. Really unsure. And I think that's also a space where I like work to get, where you don't know. I always tell myself it's either when I know it's ready, when it's like there's nothing more I can do to it, and maybe I hate it and I don't want to see it ever again. Like, that's the time to stop, and I'm taking it as far as I can. So I tried remapping them. Part of my other is every day at the studio, I would make a small hand weaving on site. So then I could trace which day had which weaving. So I sort of like made piles and tried to order them, and I placed them all like in for dates. And then I just put them on the wall, not that way. And I have this big table in my studio, so I scattered them that way. And I settled on this for presentation. And whenever I'd get to my studio, I would just add to dry on my studio wall the weaving of that day. And so as a description, there's all the ingredients that went into making the work, which I always find very important in naming and being able to know what is there and how to recreate it with some help. So writing through this artwork, I really wanted to write through something. And I used all these prints. I'll just show you details. This is also the blanket, the dice, and in my final exhibit. So they're just details from the gallery. So I used all these prints. I scanned them. This is one of my favorites. It's the ghost image with the textile that's inked flipped over. And then also a bit runny. Added string. This has discolored between when I took a picture of it and when it was made. So I've liked stability to work on, other direction. And so this was my MFA thesis paper that accompanied the exhibit, and I really wanted something readerly. And so I used all of these prints, some that I scanned two-sided, to make a flip book, and really went with a different format. And I had a section on color, which was my relationship to color, and sort of, it really made me realize how it's always there, how it's so important, but how I never place it at the center of my work. And this prompted me to, since graduation, I applied to Canada Council for a project on pigments as a relationship between weaving, printmaking, clay, and sound, which was an 11-month project, which I'm almost at the tail of. But it was really meaningful in helping me realize, and also wanting to take time to just focus on color as opposed to other work. And the other thing is that sometimes I love color tests. I come from a science background, so I can do color tests forever. But I really want to, choose with my intuition what colors I want, because there's also textile history. So this is my local print shop. I became a member very quickly in the first months that I started printmaking, just to show you the presses and the nice space. So I wanted to scale up, learn to work like much more precisely. And I got a grant to work with a printer who's also a paint maker, but a traditional printer and paint maker. So we used my pigments, but oils, which are very hard to clean and not so ecological. But so he helped me learn to work with paper, which is 30 inches by 40 inches wide, which I can barely hold when it's wet, which really had to do, you can have bad technique when you're doing small paper, you cannot have bad technique doing large. So it was really great to learn. And so for the jacquard weavings that I did for that, I always start my work by doing watercolors and then scan those, use them as compositions, and then bring it into Photoshop to add weave structures. I was looking at weave structures for historical canvases for paint making. So sort of really the relationship with the image. And then throughout the session, I re-inked my textile and then sort of give it a story, move it around, have it go be used, give it a bit of a life, sometimes re-ink it, and the details become very interesting, sometimes going to towards tearing the paper also. And I really like just the quirky details. So I kept on taking, I often take the ghost print. So the image that's left, I pass my fiber, I ink a plate, I pass my fiber through the press so it absorbs ink, and then I move my textile carefully onto a clean plate. But then sometimes I put papers to grab the ghost image, and this is the ghost image, and it sort of makes like a mirror, but it's the reverse that's together. And weaving has a lot of association to writing for me and towards text. So this is sort of like looking at the translations of text simultaneously, putting the ghosts together. And then this is also adding more color onto the cloth and some folds. So this was presented at Fiberworks, which is a textile biennial. I was very excited that they selected my prints of textiles and that I wasn't exhibiting textile. And I mean, to me, I consider it textile because it's so much in the construction of it. It's very much like it is on paper, but to me, I still consider it a textile. And so sometimes I wanted to dye papers for Chinekale. This is cochineal. And luckily, so as you can see, they're all bleeding. The quality is not what I want, but I had dropped one paper, had fallen onto my studio floor, which was very dusty and full of fiber. So then I rinsed that one, and that turned out to be the only one. So learning to dye the papers for bookmaking, you're not wetting them, so it doesn't matter. So this was a series of 20 that I did for a mixed media portfolio exchange. And I wove the fabric, and between every print, I'm taking apart the fabric and repassing it. And you can barely tell, but there's an insertion of a cochineal and an indigo dyed thread, which are added on. So it's repeating the same dyes and pigments, and you can barely see it. So there's fiber insertion in it. And this is the 20 prints. So I'm working towards doing smaller series. So I went to Vermont for a printmaking residency in October, and in September I saw that Radha Pandey, who had given the brush dyeing workshop, got a bunch of researchers on paper and natural dyes together and printed this book. So I had it shipped to Vermont, and it really changed my way of working. So I meant to bring pigments and try certain bases, but then they're using it wet, which makes a lot more sense because, I mean, in print shops, you normally have ink, so you don't have like open pigment. So then I mix my things in a safe way at my own studio and carry it, but sometimes I need to adjust the ink. So it's very harder to work. But then at the Vermont Studio Center, I had my personal studio right outside of the print shop or through the print shop. So I was able to mix my inks differently. And then I'm also dyeing papers on site. I brought my burner. So it was a really good time to spend like a full month only working on color in printmaking. And I'm just have a few photos of the studio. These are all inks. I brought some plants from my supplier Dahlia Milon in Kamouraska, but I also, because I was in the US, ordered pigments from Kama Pigments. And so I started weaving these are 18 by 24 inches. There was a smaller press. This is woven with paper fiber. So I started weaving, and these weave in the weft is paper. So then just working with paper, and the paper is made of cotton, and then in reverse, I've been ordering all types of paper fibers, which are really interesting because they don't absorb the ink the same way. And I've been like wet tearing the Chinekale papers, which is takes a very long time and is very introspective. So these are just some dyed papers, and my photographer can make everything absolutely beautiful. So there's coreopsis, buckthorn, mallow, and madder. And again, these change. This is Rudbeckia, the brown, and that's Rudbeckia with citric acid. And when I rinsed both of these, they became green. And this becomes blue, which is hollyhock. So there's the alkaline and then just the water, but I do have to rinse them. I can't preserve the color. And this is, again, this is two different. This is super thin paper weft, and this is a thicker paper raffia weft, and it's a mixed warp that I did here. The paper absorbs it. It's a green indigo pigment from Kama, and this was just a test. I was also testing a lot of different papers from different suppliers just to see. So it's really just a moment of experimentation before going towards new work. There's just a few. So this is a light, I made this just quick composition for a light fast test. So I'm going to cover half of it and put it in a south-facing window for about 6 months to see which colors are holding in different sources. And again, I'm trying different commercial bases, but with yellow ochre. And this one is definitely not working for me. And these two, I'm enjoying the definition, but I leave the prints to dry for 1-2 weeks. So then it's also to see over time how they transform. And I think this is the last print. So this is iron ochre paper. And then just really getting it's been a lot of fine tuning with moisture and wetness. So there's all the part that's very interesting in doing the weaving, but then really arriving at prints that will be stable and last a long time, and that have the right definition where I can combine color and bring color into it. I've been testing wood, dyeing some wood too for framing. So I have more tests to do with that. So if ever anyone has any interesting color information, suppliers, researchers, please do reach out. I found that I found access to a ball mill here, which was very exciting. So now I can grind my pigments much more easily. Thank you.
Thank you so much, Annie. Very exciting and beautiful practice. Congratulate Annie. And before we take questions, I also want to give a shout out to our Vice President, Paul Haft, who unfortunately was not able to be here today. Haft 2 is our sponsor for our student award, so we're very grateful that they've been able to help us support student work across Canada. So I will open the questions in the chat, or if you want to unmute, you can also ask any questions. Mantaha, go ahead.
Yeah, first of all, I wanted to say thank you to Paria and also to Annie. Very interesting. I was fascinating with both topics. For Annie, I just wanted to make a comment. It's not a question. I recently was in the Color Marketing Group Summit, and we had a speaker from the UK. Her name is Laurie Perryman, and she runs a studio called The Color of Same. And she has been, I can get you all the information outside of this. Yeah.
Okay.
And so she has been running a project called Ethical Color, which is based on finding and creating all these plant-based colors that are ethical. Okay. And so after the presentation that she made, there was an exhibit that lasted for 2 days where we could explore all these plant-based pigments. And one of the experiments that we were making with her was a cabbage ink, which is in this part here. And then we added to the cabbage ink, we added sodium bicarbonate, so it becomes green. And then we added citric acid, and it becomes pinkish, reddish, which was very interesting for us. And she's also working together with a studio in Poland. It's called Slow Painting. And they do a lot of experiments with plant-based colors. And so I thought it might be of interest to you to know about this, and I can definitely get you all the information by email if that interests you.
Yes, that would be absolutely wonderful. What I'm finding is sometimes the paper, and because I've done the cabbage on wool, it stays, but also like on wood, when I'm finishing it sometimes with soap, which is alkaline, so then it shifts my color back, and I'm like, either I need to, and I don't want to use varnish in my studio. So sometimes it's like with the next process, or when I was mixing with an oil or with like a water-based washable oil, it would shift color again, and I'm like, oh, I had the right color. So
yeah, they mentioned
and I mean, I'm not ready to
cabbage,
use.
Yeah, cabbaging is not stable. So it's good for experimenting, but up until now it hasn't been stable.
No, I mean, it's fun too. I'm working on a piece of combining stable and unstable inks and sort of having people map the color, just come and then sort of match the color with the date, and then on different days, and then they can see how some colors disappear and some are solid.
Any other questions or comments? We have actually a previous student award. I don't know if you know Vanessa Mardirosyan. She's at Concordia. She looked at sustainable color in fashion design, and you may have come across her or not, but
yeah, I've met her at Concordia, and she's come for a talk at MMAQ. But yeah, it's wonderful work. With a lot of compost and bacteria on textile. Very exciting.
Any other questions or comments? If not, please join me once again to thank the speakers and congratulate them for our student awards for 2022. And I hope everyone has a good holiday season, and join us next semester. We have CC Hart, who's a synesthete, and she was a winner of the AIC Sensing Color Award. She'll be talking about her practice as a physiotherapist with synesthesia. And I just had confirmed for February, speaking about sustainability and design, we have Ingrid Calvo-Ivanovic to speak on that topic. And then in March, we have our keynote speaker for our AGM, and we have Donald Chrétien, who's a Indigenous artist based in Ontario, who will speak about color in his work. He's got really wonderful stuff. So stay tuned to our website. And once again, thank you to the student, congratulations to the student award winners. Thank you again to HAF2 for supporting them. And we will see you next year. Bye-bye.
Thanks, everyone. That was great.